Hello everyone. Welcome back to our PlantAnatomy. In today's lecture, we'll learn all about the cell structure and its components, Part 1, in a simple and easy-to-understand way. So let's begin. The cell, the protoplast, the basic unit of which organisms are constructed are the cells. Robert Hooke, in 1665, first used the term cellula, a small apartment for the small cavities surrounded by walls in cork. Later, he observed cells in other plant tissues and noticed that the cavities of the living cells are filled with juice. Later, many observations of protozoa and animal tissues led to the conclusion that cells also existed in animals. In 1838, the German zoologist Theodor Schwann and the Belgian botanist Matthias Jacob Schleiden established cell theory, stating that all organisms are composed of cells and the products of cells. The cells are building blocks of life, and the life continuity of the cells is maintained by the division of cells. The substance within the cell received the name protoplasm, from the Greek words for first and molded, meaning living matter in its simplest form. It is colloidal, semi-fluid substance of changing consistency. In 1880, Hanstein introduced the term protoplast to indicate unit of protoplasm found in a single cell. He also suggested that the term protoplast should be used instead of the term cell. But this suggestion is not generally accepted. In plants, the term cell includes the protoplast together with the wall. Nowadays, the protoplast is the protoplasm with non-living inclusions, excluding cell wall. The protoplast of a plant cell secretes about itself a cell wall of cellulose. All the protoplast, plus the cell wall, is the plant cell, the structural and functional unit of the plant body. For a long time, the cell wall was considered as an ergastic substance, but recent researches have shown that organic unity exists between the protoplast and the wall, and the two together form a single biological unit. Robert Brown, in 1831, discovered nucleus in an epidermal cell of an orchid plant. In 1846, Hugo von Mohl distinguished between the protoplasm and the cell sap. And in 1862, Kolliker introduced the term cytoplasm, the material surrounding the nucleus. Two types of cells, the prokaryotic and the eukaryotic cells, are recognized on the basis of internal organization. The eukaryotic cells have a membrane-bound nucleus and other organelle, and is of animals and plants except for blue-green algae and bacteria, while the prokaryotic cells lack these membranes and are found in blue-green algae. The cell usually contains a single nucleus, but in some cells, e.g., in sieve elements of the phloem, the nucleus is generally absent from the mature cells. There are some cells which have many nuclei, multinucleate. For example, in some latticifers, a multinucleated cell can comprise an entire organism, as in some fungi and algae, or multinucleated cells may be a transitory stage in the development of a tissue, for example, in the endosperm of many plants. The view had been advanced that in some multinucleate structures, each nucleus and the adjacent cytoplasm represent a cell. And the whole structure is an aggregation of protoplasmic units termed coenocyte, from Greek koino equals in common, plus cyte equals a vessel. Two theories exist which deal with the relation of the entire organism and the single cell. According to the cell theory, the organism consists of a complex of cells, each of which plays role in determining the nature of the organism. The second theory, known as organismal theory, gives less importance to the individual cell and mainly stresses the unity of the protoplasmic mass of the entire organism. According to this theory, The organism as a unit determines the nature of the cells. A plant cell is distinguished from animal cell in the presence of I. Cell wall, 2. Plastids, and 3. Vacuoles. Size and shape. Mature cells vary in size and shape. Cells may be ellipsoidal, ovate, cylindrical, flattened, prism-like, star-shaped, fiber-like, and lobed. Parenchyma cells are usually 10 to 100 micron in diameter, but larger cells are found in fleshy fruits and pith of the stem. Fibers are usually about 1 to 8 millimeters long, but fibers as long as 55 centimeters are also found. In meristematic regions, the cells with primary walls tend to assume the smallest possible surface area, i.e., the form of a sphere. After mitotic forces act that tend to impart to the cell a spherical form, but they become polyhedral in shape due to mutual pressure, as there are no intercellular spaces in the meristem. The basic shape of the cell is a 14-faced polyhedron, but cells with 12, 13, 15, 16 or more faces are also found. Most of the faces of the cell wall are pentagonal, but tetragonal and hexagonal faces are also found. Due to increase in cell volume during growth, the number of wall faces increase above 14, making it impossible for all the sides to remain in contact with all the sides of the neighboring cell. And so intercellular spaces develop. In some tissues, these spaces are quite large and are known as air spaces, ducts, etc. Such spaces can develop in two ways. A. By separation of neighboring cell walls, as in the development of the resin ducts in Pinus. This type of development is called schizogenous development. B. By the disintegration of the cell in the place where the space develops, as in oil cavities in the peel of citrus fruits. This type of spaces are formed by lysigenous development. In some cases, as in the protoxylem, the spaces are formed by these two methods together, and this type of development is called schizo-lysigenous development. The cells show two types of relationships with other cells during the growth. When a group of cells grows together uniformly, the cells of the group take up different positions and areas of contact are formed between the neighboring cell walls do not change, and no new of contact. In many cases, hit-free tips may grow along walls of the neighboring cells and form new areas, fibers, etc. This type of growth is called intrusive growth and takes place in many elongating organization of cell. The following components are commonly recognized in the protoplast of plant cells. Protoplasmic components. Non-protoplasmic components. The thin membrane enclosing the protoplasm. I. Plasmalemma. Protoplasmic contents are I. Plasmalemma, material enclosing the other protoplasmic bodies, i.e. plastids and mitochondria, and non-protoplasmic materials, various granules, and membrane systems such as ribosomes, lysosomes, etc. 3. Nucleus. A body considered to be the center of synthetic and regulatory activity and the seat of hereditary units. 2. Non-protoplasmic components. The non-protoplasmic components of the cell includes vacuoles, the cavities with cell sap bounded by a membrane, the tonoplast, and contain more or less solid inclusions such as crystals, starch grains, and oil droplets. 2. Ergastic substances, from Greek erg work, the non-protoplasmic substances, secreted and maintained by protoplasm, present in the cytoplasm. 3. Cell wall. It is considered to be composed of ergastic substances, but are deposited on its surface. It is boundary wall of a plant body. 1. Protoplasmic components. I. Plasmalemma, cell membrane. In all the cells, the protoplasm is enclosed by a thin, film-like, pliable membrane called plasmalemma or cell membrane. Eukaryotic cells also possess intracellular membranes that bound the vacuoles, tonoplast, and organelle. Plasmalemma consists of proteins and lipids, but in certain cases, polysaccharides, RNA, and DNA have also been found. 3 different kinds of proteins are found in the plasmalemma. These are I. Structural proteins. These form backbone of the membrane. These are lipofloic in nature. 2. Enzymatic proteins. These proteins specify the physiological function of the membrane. For example, in mitochondrial membranes, cytochrome oxidases are present, which are concerned with respiration. 3. Carrier proteins. These proteins are not well defined, but have been reported in Salmonella typhimurium, Escherichia coli, wherein these act as sulfate carrier and glucose binding factor, respectively. The lipids of plasmalemma are 5 different types of phospholipids. In addition, these phospholipids have either choline, inositol, ethanolamine, or serine. The choline-containing probiotics. Share it with your friends, something Dory, and don't forget to subscribe for more easy biology lectures. Answer the following questions. Statements from section, what is cell? 2. What are protoplasm? 3. What are eukaryotic and prokaryotic cells? If you know the answer of the following question, then write in comment section. If you have any question, then ask in comment section. Thanks for watching, and see you in the next class.